Here we are going to discuss an image-based question where a histological picture is given to us, and the question that is asked is identify the given histological diagram. So whenever we look into any histological picture, the most important features we have to make out are the epithelium and the underlying connective tissue. So here also in the given image you can see the epithelium. This is the topmost layer of the epithelium and you have the basal layer here. So here you can see that the basal layer is extending into the underlying connective tissue in the form of retipex. So here you can see that the retipex appear more prominent and they are nothing but the extensions of the epithelium into the underlying connective tissues. Therefore, this is the epithelium and the retipex appear very prominent in this picture, and the portion beneath the epithelium is the connective tissue. And the portion that is above the epithelium is the keratin layer. So here you can make out epithelium with the retipex and the underlying connective tissue. So with this basic knowledge, now let's look into the options given to us. They are lichen planus, pemphigus, epidermolysis bullosa and pemphigoid. All the four conditions are mucocutaneous disorders, and the characteristic histological findings we should be aware of in order to come to the diagnosis. So in case of lichen planus, the characteristic feature one would see are sawtooth retipex, subepithelial band of T lymphocytes, and then you will find degeneration of the basal cells, and this degeneration of the basal cells will result in cytoid or civate or colloid bodies, whereas it results or it weakens the epithelial connective tissue interface, which result in Max-Joseph cleft spaces, Max-Joseph cleft or Max-Joseph spaces. So these are important histological findings that is characteristic to the lichen planus. Apart from that, we'll also find hyperkeratosis and acanthosis in case of lichen planus. Next comes the pemphigus. It is an autoimmune blistering disease. So here the most important characteristic feature is the suprabasilar split. And in case of pemphigoid, it is the subepithelial split. So therefore, pemphigus is differentiated from pemphigoid based upon the level of split that is seen. So the vesicle formation is at the suprabasal level in case of pemphigus, whereas subepithelially in case of pemphigoid. When you come to epidermolysis bullosa, here you can see that both intraepithelial as well as subepithelial split are encountered in different types of epidermolysis bullosa. So with this basic findings, now let's look into the given image. So which histological finding match the findings in the given image. So here you can see, as I have mentioned, the retipex are going to be the prominent one. So therefore you can see that the retipex are taking up the shape like a sawtooth projecting into the underlying connective tissue. So therefore sawtooth retipex matches here. And then you can find a subepithelial band of T lymphocytes. So just beneath the epithelium, you can find a band of lymphocytic infiltration as a basophilic appearance, that is nothing but T lymphocytic band. So it is an autoimmune disease. So the T lymphocytes migrate towards the epithelium against the basal keratinocytes, and that results in formation of subepithelial T lymphocytic band. Apart from that, here you can also make out that there is a thick keratin layer known as hyperkeratosis. So therefore, these are the important findings, and these findings help us favor more strongly the lichen planus to be the answer for this question. So, but here also we have to rule out the other things, that are we finding any suprabasilar split or subepithelial split in this case. So suprabasilar or subepithelial split is not found, whereas all the layers of epithelium remain intact here. Here you can see that within the epithelium there is infiltration of T lymphocyte intraepithelially also. This is again a finding that favors lichen planus. And then we don't find the level of split. There is no suprabasilar vesicle formation or subepithelial split that is evident here, whereas the most characteristic finding are the hyperkeratosis, sawtooth retipex and subepithelial T lymphocytic band that favors lichen planus to be the most suitable answer here. So now let's discuss the various other features also and compare the histological picture for a better idea. So here you can see the lichen planus, as I have already mentioned, the low power shows all the features such as hyperkeratosis, such as subepithelial band of T lymphocyte. Here you can see the sawtooth retipex that is evidently seen here. And when you look into the second image, that is the high power, here you can see the sawtooth retipex in the high power, and you can see the T lymphocytes, subepithelial band of T lymphocytes, and here you can also find out that there is degeneration of the basal cell with the infiltration of T lymphocytes. The basal cell undergoes keratinocyte apoptosis that results in the degeneration of the layer or epithelial infiltration of the T lymphocyte, is evident in the high power image. Whereas when you come to pemphigus vulgaris, we have to know that there is suprabasilar vesicle formation that results in acantholysis of these cells. It is loss of cohesion between the layers of the epithelial cells which result in a suprabasilar split. Here you have to notice that the basal layer remains intact. And within the suprabasilar split or the vesicles, the epithelial cells come to lie free within the vesicular space, and they are known as Zank cells. And it is characteristic of pemphigus vulgaris. In the first image, if you see, you can see that the basal layer is intact. So there is no degeneration or any changes in the basal layer, whereas in the suprabasal layer, you can find that there is a cleft or there is a split with the epithelial cells lying free within the space, and these are known as Zank cells. So this Zank cells forms a unique feature in case of pemphigus vulgaris. And looking into the second image, which is a high power, here again you can see that the basal layer is intact, whereas in the suprabasal layer you can find out there is a split. This is the suprabasilar split, and here you can see various epithelial cells lying free within the space, and these are nothing but the Zank cells. Next, looking into pemphigoid, as I have mentioned earlier, you will find a subepithelial split in case of pemphigoid. So here you can see that the basement membrane and then the underlying connective tissue, between which you can find out there is a split. So this is known as subepithelial split, that is between the basement membrane and the connective tissue. So the pemphigoid shows a subepithelial split which is different or contrast to the suprabasilar split found in pemphigus vulgaris. And next, coming to epidermolysis bullosa, as I have already mentioned, it shows different types. It is both intraepithelial as well as subepithelial split based upon the different forms of epidermolysis bullosa. Whereas if it is epidermolysis bullosa simplex, you can find an intraepithelial split. Whereas in case of the other two types, that is the junctional type or dystrophic type, and the third, that is the recently categorized Kindler syndrome. So all these three conditions or the three types of epidermolysis bullosa will show subepithelial split. So therefore, intraepithelial and subepithelial split can be encountered in cases of epidermolysis bullosa. So looking into the picture, here you can see that the split is present subepithelially. Subepithelial split is encountered here. So therefore, these are some of the characteristic findings we should be knowing in case of pemphigus, pemphigoid, epidermolysis bullosa and lichen planus. So based on this given picture, we can make out that it shows hyperkeratosis, it shows sawtooth retipex, it shows subepithelial band of T lymphocyte, even the T lymphocyte penetrating into the epithelium, that is intraepithelial infiltration of T lymphocyte is found here. So all these histological features are strongly suggestive of lichen planus as the most appropriate answer for this question.